Welcome to Almost Here, Round the Corner Future Technology podcasts with Richard Jacobs. Future technologies poised to transform our lives for better or worse are the focus of this podcast. Almost here means these technologies are now here and starting to be used, or just around the corner, from Bitcoin to artificial intelligence, 3D printing, blockchain, virtual reality, and more.
Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast. My guest today is Emeran, MD. He's a professor at UCLA, and he's the author of the Mind-Gut Connection. So, Emeran, thank you for coming. I appreciate it.
Thanks for inviting me to be on the show.
Yeah, what gave you the premise for your book? How did you discover there is a connection between the mind and the gut, and do most people know there is such a thing?
Well, I've spent pretty much the better part of my career studying the brain-gut interactions. I've always been interested in brain-body interactions from a clinical, but more from a research standpoint, and pursued that first in terms of communication between the brain and the cardiovascular system, the heart, and then really based on my decision to become a gastroenterologist, decided to shift the focus on the brain-gut connection. And in terms of the microbiome, that has not been something that was on my horizon until I would say about 6-7 years ago, when the first papers came out that suggested that there is this connection of first studies been done in mice, and that remains the same, that the majority of studies of really breakthrough studies have been done in experimental animals. And so when I saw these findings, I said, we have to sort of explore this, and reluctantly, really, have to say, got into that. And we did a study that changed my mind, which was basically an intervention study with a very simple concept that you feed healthy people a probiotic cocktail containing yogurt for 4 weeks, and then you study them before and after in terms of symptoms and particularly brain function, various measures of brain function. And that study came out positive to my big surprise. And ever since then, we have included the gut microbiome as a major readout in all of our studies.
Well, let's wake people up. What are some facts or statistics that really surprised you in your research? Maybe just a few of them, and then we can dive in.
Well, in my own, so the biggest surprise was really at the beginning, because I was a huge skeptic that you could change emotional behaviors, as has been shown in these animal studies. And there's a lot of concerns about the relevance of these early studies, because they were all done in mouse models that are not really translatable to humans, like these germ-free animals. So to me, that we could demonstrate there is if we perturb the homeostasis of the gut with this probiotic cocktail, that we would see a signal in the brain of that change, basically demonstrating that this communication from the gut to the brain, from the gut microbes to the brain, must exist in humans as well. A big difference was in that we didn't really get any behavioral or clinical readouts in our human population. So this was purely a brain finding, and it was actually not associated with a change in the microbial composition, but previous study had shown that exactly the same intervention led to a change in the metabolite profile that the microbes generate. And so, this to me was really the biggest surprise. And, we have continued to do studies in that direction that, they're still surprising that, there's correlations between the architecture of the gut microbiome and some features of the brain in terms of structural and functional properties. And most recently, that was another big surprise, I have to say, from in the human domain, that we looked at the gut microbiome and symptoms and the brain in patients with irritable bowel syndrome that underwent uh, a trial of a course of cognitive behavioral therapy. And I mean, again, to our surprise, was that there was, even after um, a relatively short course of intervention, like a 4 session cognitive behavioral therapy, there were changes at the brain level in terms of connectivity of, in this case, brainstem areas with other brain networks, but also that the microbes were somehow involved in this. The composition at baseline of the microbes, the relative abundances, um, seem to predict the responders to this therapy. So responders were defined by symptoms measured with validated symptom severity scale for IBS. Yeah, so we found
Wait,
that.
you mean you could tell if a certain person with IBS was going to respond to a treatment based on their microbes or the metabolites of their microbes, or I'm not
No,
sure what you're saying.
this was really based on the relative abundance of the microbes, and this was a big surprise because we did this study really hypothesizing that there would be a change in the microbes, both the metabolites and their relative abundances, with the intervention, and we thought this would basically prove that big influence on microbial composition and function um, is being exerted by the brain, and if you modify that with a purely brain-based, brain-targeted therapy, you would see that signal in the microbial domain as well. And
Well, what was the just to put a little context? What was the intervention that was proposed, and what did you think it would do, and what did it actually do?
well, the intervention, so cognitive behavioral therapy is a fairly standardized intervention. There have been previous studies in different patient populations, including IBS. There's um, a short-term version of that therapy. So typically, it's 10 to 15 sessions that a patient has to go and see a specialized therapist. In this trial, this was a trial by Jeff Lackner at the University of Buffalo. He compared the response, the clinical response, to three types of things: an education control, a short-term patient-focused cognitive behavioral therapy with 4 sessions, and a traditional cognitive behavioral therapy trial with I've forgotten how many. I think it was 10 or 15 sessions. And what that studies, so this was in several hundred patients, what they found that the two cognitive behavioral therapy interventions had a significant benefit on the symptoms of IBS, confirming earlier studies. What was interesting that this short-term therapy was as effective as the traditional, more prolonged intervention. And what it is basically patients are. So this is not regular psychotherapy. This is teaching patients to change their coping skills, to change their stress responsiveness, to change, for example, one characteristic trait of IBS and other chronic pain patients is catastrophizing, essentially assuming worst case outcomes occurring with a very high likelihood. So these are all the components that are retrained by this kind of therapy. And there's also relaxation exercises. But anyway, so this is clearly something, an intervention that does not involve the gut as a target. So there's no dietary recommendations. There's typically no other lifestyle changes that are part of the intervention. And so we thought it's a perfect tool to demonstrate that the brain has a major influence on microbial composition, and that has always been. I mean, there's many studies in animals, in humans, in monkeys, that different types of stress will change the gut microbial composition. Typically, there's a decrease in the relative abundance of lactobacilli, and there's also been shown that this has effects on tryptophan metabolism in the gut. So there's pretty good evidence for that. Most people, certainly in the drug development or in the therapeutic area, don't think so much about this top-down modulation. Everybody wants to change the gut microbiome and then expect, behavioral therapeutic changes. This was clearly
Well,
tested the other.
how do you think that the mind? I mean, I'll just step through it as I don't know. This is my assumption, but so let's say you're stressed out a lot, therefore your cortisol elevates and other chemicals elevate, and it changes the environment in which your gut bacteria live, causing certain ones to be pressured to die out and other ones to flourish. Is that kind of the mechanism by which your gut microbiome is altered, or is there a more direct, connection between the mind and the gut?
Well, there's at least two ways. I mean, so the HPA axis and cortisol has not really been shown that playing an important role, but the other component of the stress response, the autonomic nervous system, sympathetic and parasympathetic branches of the autonomic nervous system, which innervate the entire gut and have previously been shown in many studies, animals and humans, can modulate gut activity. Pretty much all the gut functions, from secretion to motility to regional transit to um, fluid secretion, mucus secretion. So all these gut functions are under autonomic nervous system control. So if you have an autonomic nervous system that chronically hyperreacts to events that the individual perceives as stressful, then you have to, you have to assume that this will affect the habitat and the environment for the microbes. And that has been shown for acute stressors and chronic stressors, that they all have this effect. To me, it's always interesting how relatively little attention that part of the mind gut microbiome interactions gets. It's the bias is clearly on the other side that can be influenced with medications and diet and
What's the mechanism? If you was to, in plain speak, what's the mechanism by which your thoughts and your feelings affect your microbiome?
two ways. The one that I just mentioned, that through the autonomic nervous system, it changes the environment that these microbes live in.
How would it change the environment? What's an example of how it could change the environment?
All the factors that I mentioned before:
Okay.
regional transit, motility, secretion. There's secretion of antimicrobial peptides. There's secretion of mucus, of fluid. There's changes in blood flow. All of these fundamentally change that habitat, and and most likely based on. I mean, this goes sort of too far, really. So it's been studied that different kinds of emotions, like anger, fear, um, anxiety, have different patterns that they change the gut environment. So some slow gastric emptying, others speed up gastric emptying, others increase contractions of the sigmoid colon, others inhibit the contractions. So you could almost hypothesize that different types of emotions, chronic emotions, create different environments for the microbes, and that may then be translated into different metabolomic profiles that these microbes generate. But there's also one other, direct pathway, and that is that norepinephrine, so one of the main stress mediators that goes up in the blood and drives up heart rate and blood pressure. It's been shown that norepinephrine somehow gets into the lumen, into the inside of the gut, and interacts with receptors that are very homologous to to the human, to the mammalian the adrenergic receptor, and that influences the bacterial behavior. It makes them more aggressive, more virulent, changes their gene expression profile and their behavior. And so you have really two ways. You have a direct way through this stress mediator, and you have the indirect way by changing the environment. So in any case, you would assume a pretty profound change occurs in individuals that are chronically stressed, or are suffering from chronic mental disorders. And it's so stress and anxiety are big components of a whole range of psychiatric disorders. So depression, obviously anxiety disorders, autism spectrum, anorexia. So in all these populations, people have found in cross-sectional studies that there's changes in gut microbial composition and function. but we don't really know if that could be a consequence of the underlying brain disorder, or if these altered microbial features, microbiome features, play a major role in the generation of symptoms.
Are you
Again,
able to establish how fast someone's mindset can affect their gut, or is this just by definition a long-term type of process?
no, if studies have been done with acute stressors in humans, noise or um, listening to two different sounds in both ears. So there's a whole range of these acute laboratory stressors, and many of those have shown an acute effect on, for example, the motility of the GI tract. There's also some classic older studies where people with that had stomas, basically where the gut was surgically, because of an accident or because of tumor removal, the opening of the gut was visible from the outside, so you could actually observe it from the outside. And
Right.
with acute emotional changes in the blood flow and the secretion in these parts of the GI tract were observed. So this happens on an acute level in everybody. I don't know if it happens if you get upset for 5 minutes because something happened in the environment, you're stuck in traffic. But certainly, if you have a day that you perceive as stressful, almost certainly that will affect your gut and your microbes in it.
Mm. Mm. I would think that depression and anxiety are huge, and there's going to be some big connection here. Do you know? Have you looked at those conditions at all, or are you not there yet? Is there too much to look at?
No, so we personally have not looked at it, but there's several published papers now. I think there's 3 or 4 with depression. Typically, these are cross-sectional studies, so you can only talk about associations, not causation. And all 3 studies have shown that there is a difference in the relative abundance and also difference in the metabolites that the gut microbiome produces in patients with major depressive disorders. And in a couple of these studies, investigators have taken stool samples from these patients and put them into germ-free mice or rats where the microbiome has been suppressed with antibiotics. And these animals then adapt some kind of altered behavior. It's not always depression-like behavior, but there's an alteration in the behavior of these animals. And there's an alteration when they looked at the gut microbiome after transplant. They showed similar changes as the human donor had in their gut microbiome. So the same thing is true about. I mean, there's a whole range of these disorders now. There's autism spectrum, there's Parkinson's, there's depression. We have not looked at those ourselves, but there is a growing literature which, I mean, you can always criticize things it's not controlled for dietary differences. The main concern is there so far have not been studies that really prove the causality. As I mentioned earlier, all these observations in theory could be related to the brain sending different signals to the gut in these patient populations. And so the model, the model that we have proposed to kind of explain that really for all these brain gut microbiome disorders is that it's really a circular process which can be initiated by an influence of the brain on the microbes, changes the metabolites. Metabolites feed back to the brain and either reinforce or amplify certain features, and then the brain sends back the signal to the gut. So it's a circular system. You can't really say it's either caused from the gut or from the brain. So that's the model that that we think fits best most of the published data to date.
Is there one way you think that's acts faster or is more powerful? I mean, do you see it as a cooperative thing between mind and gut, or as a battle? Which side would you pick? I guess if you had to pick a side,
No, in a healthy person, clearly it's a symbiotic system. It's beneficial the brain gets important information from the gut, and the microbes, have an optimal environment to live in, and they have adapted to this environment. But in disease states, then, I mean, I personally still think a lot of, and I mean, I may be wrong. It's just I think that a lot of these traditional brain disorders, either neurology or in psychiatry, have a very prominent brain component that may be at the beginning of it, and but then change the whole system over time, and then both components become maybe equally important to maintain the chronicity of the disease. I mean, there may be one example to that statement. So I mentioned Parkinson's disease briefly. So in Parkinson's disease, there's some very intriguing findings that these degenerative um, uh, these, these characteristic degenerative changes that have been described in the brain, postmortem brain of Parkinson's patients, that they actually occur much earlier, up to 20 years earlier in the nervous system of the gut, the so-called enteric nervous system, in a subset of patients, and also that these, so they're called Lewy bodies, that they may actually move through the vagus nerve from the gut to the brain, first brain stem, and then the brain, and that this process, as I said, could take up to 20 years before the classic neurological symptoms appear. So that's, I think in that case, that's kind of well, I mean, it's not really an exception, because once these degenerative changes reach the brain, it will change the autonomic function, the vagal output back to the gut, and constipation is an important feature of Parkinson's. We don't know if that constipation is a consequence of the degenerative changes in the enteric nervous system or in the autonomic nervous system, but there's clearly that feedback as well. So now, what starts it in the gut, we don't really know. There's cross-sectional studies that show there's an alteration in the gut microbiome in patients with Parkinson's, but it's not known if that's due. It could be a variety of factors. It could be because of the stress of this individual, because of the slower transit, because the whole motility is impaired by the enteric nervous system degeneration, and so the microbial changes could all be secondary, or they could be primary.
Have you observed bacteria from the gut getting through the blood-brain barrier, or in the vicinity of the brain, or in the brain cavity somehow?
No. So there's two answers. A very recent study showed was just presented a couple of weeks ago at the neuroscience meeting in San Diego. So people were able to identify microbes in the brain of post-mortem, brains. So these were people that were otherwise healthy, had no brain disease, but they had microbes could be detected in the brain. So it's one study, needs to be confirmed. Obviously, that's not a contamination, but it's conceivable that just like in the placenta, which always has been considered sterile, and we now know there's some bacteria, that the same thing happens in the brain. So the other answer to this is, so the bacteria by themselves are unlikely to actually mediate this gut microbiome brain communication. It's their metabolites or immune signals or nerve signals that are triggered in the gut by the microbes and then affect the nervous system. So I think to answer this, preliminary, tentative, there may be some microbes in the brain. We don't know what they do, if they have any negative effect, but much more likely that microbes in the gut produce metabolites, so-called neuroactive metabolites that can go to the brain, can cross the blood-brain barrier, or that act on the vagus nerve to send signals to the brain, or they interact with the immune system, which we do know, and thereby changing immune signaling to the brain.
So what do you think is what's an easier way to study what's going on in someone's gut? Is it looking at the bacteria themselves or the metabolites themselves? Do you really need to look at both in order to get an understanding of what's going on?
So the field is clearly moving towards characterizing the function, which means metabolites or gene expression profiles. So there's all these omics, metabolomics, metabolites, proteomics, the proteins that are being the production is influenced by the microbes. Metatranscriptomics, so which genes are being transcribed. The research goes rapidly in that direction. So the actual number and relative abundances, they're still important. So, for example, a very important concept in gut microbiome research in general is the ecological view of the microbiome, that it's a big ecosystem, and we know a lot about ecosystem behavior. So the diversity of an ecosystem and the relative abundances of elements in an ecosystem play a major role in determining the resilience and the stability of that ecosystem. So what we see in Western populations, that there has been this progressive decrease in the diversity in all measures of diversity of the gut microbiome. People think like up to 40% reduction in the diversity, and decreased diversity has been taken as a predictor of that these microbiomes are more vulnerable to insults, be it infections, being reactions to different diets. So that's where the numbers and like this technique, the 16S sequencing, is still important. But actually, to determine the mechanisms by which the microbes affect the brain and other organ functions, it's really these other techniques that are so rapidly taking over. Most likely, we'll see both of these or a combination of these techniques continue to be used.
Yeah, it's amazing. I didn't realize so much was involved. Wow. So what in particular, where do you want to take the research, or where are you taking the research in particular?
Well, so since I'm a gastroenterologist and with a long-standing interest in so-called functional brain-gut disorders, which are now renamed really disorders of brain-gut interactions. So we want to essentially establish, is there a causal relationship between altered gut microbes and altered brain activity, brain function, behavioral changes such as increased stress responsiveness, increased pain sensitivity. So this is one thing, and we're sort of pursuing this with sort of following the big data paradigm. We have, by now we have like 1,000 patients. But we have all the data from the brain, behavior, clinical microbiome, and we want to see, which is likely, are there subgroups of patients that may have the same symptoms, but the symptoms are generated by different types of microbial disturbances. So that's one. Another thing that continue to be very interested in pursue these mind-based therapies. How can you change a dysregulation of the gut microbiome simply by teaching the brain to send different signals. And the third one is dietary intervention. So I developed a really strong interest in the role of diet in influencing gut microbial diversity and function. And it's interesting that I think this was not anticipated when microbiome science sort of became so prominent and grew exponentially, that it would require that nutritional sciences are going to become much stricter and much more scientific. And I think from all the evidence that we currently have is that, instead of having these culture wars over what's the best diet, if you go by the microbiome, what's best for the human gut microbiome, and what's best supported by epidemiological studies about health benefits, there's no question that predominantly plant-based diets, win in this. And it's not. There's a few exceptions for temporary changes in these dietary patterns that are optimal. So in children with therapy refractory seizures, epileptic seizures, a ketogenic diet seems to be beneficial. certainly not as a long-term
Right.
therapy, but clearly as a therapeutic intervention. Some people think that a diet that's low in 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 certain complex fibers, the so-called FODMAP diet, is beneficial for IBS patients. I'm sort of skeptical of this long-term benefits of that intervention, but overall, I think, and this has really been an influence, a very strong influence on my own thinking that
Hmm.
that diet, well, I mean, diet and mind are are the two factors I think that are currently, I mean, this may all change in 10, 20 years, but currently the most
Right.
effective therapeutic intervention.
Well, what about fecal transplants? Those for a while seem to be in the news a lot. Now it's going quiet.
Yeah, the reason it's gone quiet because it's kind of a miracle therapy if you have C. difficile colitis, an overgrowth of a particular organism, this Clostridium difficile. Then this is by far the most effective therapy. But unfortunately, despite multiple trials going on in IBS, in inflammatory bowel disease, and autism, there's really not been a major demonstration that would say this works equally well. And I mean, the main reason for that, in my opinion, is that even an altered and a dysfunctional microbiome is still very stable. Because it's an ecosystem. It's not, and it will defend itself to any attempts to change it. Like with a fecal microbial transplant, you would want to get rid of the old structure and replace it with a new one. People have tried to combine this with doing first antibiotic treatments to wipe out the dysfunctional um, microbiome, and then do the transplant. People doing repeated transplants over several weeks. So there's many things going on, but the the short story is that so far the success from the C. difficile colitis treatment has not been able to be replicated in any other disease.
Well, if it's hard to, if it's hard to change the microbiome, is it just something that happens slowly over time? Let's say I change my diet or I eat a bad diet for a period of time. How long will it take my microbiome to react? Is there any data or studies that looks at that?
Yeah, so that can happen fairly quickly. And what changes the most is really it's the relative abundance and the metabolites that are being produced. So the metabolites, obviously, if you feed your microbes something different, they will change producing different metabolites. The Typically, if you stop that particular diet, it will slip back to the original, to the default um, configuration that was programmed during the first 3 years of your life. So we have this period where the basic architecture is established, 3 years, and then it's fairly stable for the rest of our lives. But what you can do is you can manipulate it and modulate it by dietary changes, by taking antibiotics. Typically, it always bounces back to its original state when you stop these interventions. There's some exceptions, obviously, like the C. difficile colitis happens in people that um, get antibiotics, but it's a small subset of patients where that happens. But normally, it's a very stable system, and what happens in a disease state, it just assumes another stable state. It doesn't completely lose its stability landscape. It just switches to another stable state, which is the disease state, and but then it bounces back to the original state if the intervention stops. Another point is important to remember. So we assume that I mentioned this, the plant-based diet leads to a high short-chain fatty acid production by microbes, and that's beneficial for our gut and most of our functions of our body and brain.
Hmm.
And um, that ability to generate these short-chain fatty acids in Western countries is significantly lower than, for example, in populations, populations have been studied in Africa and South America that eat kind of hunter-gatherer diets, and it doesn't seem to be possible to get back, no matter how many vegetables and fruits you eat, to get back to that same diversity that these people have, these tribes have. So our capacity, our bandwidth to return to the optimal state is limited, and this is something that most investigators think is a consequence of what happens early in life. That the exposure to antibiotics, both the mother, the infant, the dietary patterns of the pregnant mother, that all they have the ability to program this early life um, microbiome, and which then stays with us at a compromised level. So if you want to do something dramatically, some people propose that you would have to do a fecal microbial transplant, either from a super healthy individual or one of these primal people, these hunter-gatherers, and then it's possible then that this infant, this person, for the rest of their lives would have the optimal diversity of a microbiome. Um, so if it's going to come to that, if people, I mean, there's obviously risks involved with this as well, but I think the interventions, both dietary, medication-wise, basically a reduction of medications, and potentially fecal microbial transplants or administrations of microbes that we have lost in the Western world. Um, if you do this in the first 3 years of life, you could have a dramatic, long-lasting effect.
So, a purely dietary intervention, I mean, what are some basic parameters on how long you think it might take to have an effect on the microbiome in a way that you want and one that'll be sustained?
Well, in an adult, so let's assume the example. Somebody grows up on fast food, then somehow has this eureka moment to say, okay, now I'm going to go on the healthiest Mediterranean, largely plant-based diet. It will change fairly quickly. It will stay optimal for as long as you eat this diet. If that individual goes back to its original fast food diet, it will just switch back to the original. So it's not, it's not maintained. Um, so our recommendations to people, dietary wise, is these are lifelong changes you have to make. This is not something that you can temporarily fix. Eating all kinds of probiotics or prebiotics or a lot of fiber. This has to become a lifestyle, a long lasting lifestyle change. It's not
Okay.
necessarily what people like to hear. Everybody wants to have these quick fixes, but it's kind of similar when you think about all these failed diet therapies for obesity, which clearly involve to a certain degree the microbiome as well. And some of them are quite successful as long as you stick with them. But the minute people stop, it bounces back and actually overshoots the baseline level of obesity. If you stop it and go back to your regular diet.
Well, you said that the microbiome is resistant. It gets into a certain state and it wants to stay there. Maybe just a few comments on, what are the main modulators of keeping the microbiome in a certain state versus another? And are there sweet spots that it can drop into where it'll be more stable than in other, conditions?
Yeah, this is this whole concept about the stability landscape of ecosystems. So in a healthy person, the state it is in is so the deepest and the most resistant to change. If you are genetically vulnerable or had a lot of negative things happen early on in life during the programming phase, that initial depth of that stability valley is is much smaller, and it's much easier to be perturbed and assume another stability state. And for most, you know, for chronic diseases, typically that chronic disease stability state is fairly resistant to change. That's why it's chronic. For other diseases, acute, transient, short-term diseases, it bounces back to the original healthy state. So, I mean, you could look at the whole, the whole landscape in which health and disease is situated. It's this landscape with mountains and valleys, somewhat deeper, somewhat shallower, and the deeper and the more stable the original, the baseline state is, the much less likely is that you're going to be switched to any of these other either transient or chronic states of system dysfunction.
Well, I would think that we would want to study, ways to disrupt an unhealthy microbiome and to get it into a healthier state, and then ways to keep it there.
Yeah, and that's
about things.
So we talked a little bit about this. So one attempt for that would be aggressive treatment with broad spectrum antibiotics to knock out pretty much the or to diminish the diversity and stability of your disease state, and then come in either with particular diet or particular microbial transplant. So that's clearly an avenue I think people will have taken in autism spectrum. The danger is, you could cause a lot of harm. I mean, for a person to undergo long-term broad-spectrum antibiotic treatment to knock out your microbiome, I mean, you could develop all kinds of other things in this process. But I think we will learn.
Targeted? Is there yet anyone studying targeted microbe killers or specific microbes in the gut?
Yes, that's another approach. So these specific microcidal agents, that approach has been used in the oral microbiome. So there's a particular organism that greatly increases the risk for tooth decay and gum disease. And there's an investigator formerly at UCLA who has developed a compound that can knock out that particular organism. And so that's another. We haven't found any of these types of specific organisms in the gut, so you can't say in obesity there's one that is bad, and if you get rid of this, then you become lean again, or so that has not. This could also, I mean, this is quite. It's a potential scenario, that you would basically engineer the adult microbiome in a way that. I mean, there's also this whole thing about the stability of the microbiome is called colonization resistance. So it's so stable that it won't let other organisms set foot inside of that ecosystem. So people have worked on techniques and strategies to overcome that selectively for a particular organism, and so that may be another approach. That we learn techniques to selectively break into this very controlled environment by inserting certain organisms. But I mean, my opinion, we're in such an early stage of this field, even though millions of money has gone in from investors and all the startups. But I think like any of these strategies that I mentioned, other than what's most effective now, which is very dietary, this will take time, and this will because we still know so little about that system that manipulating it or knocking out particular organisms, it always has the risk of that. The Australia example where they let rabbits loose at some point and not thinking about what this would do to the ecosystem. And I think that's the danger that many approaches that do not follow this ecological viewpoint of the microbiome, that this is the risk that they run. This is not traditional microbiology. This is not traditional pharmacology. This is really ecosystem or systems biology science.
That makes sense, yeah. Okay. Well, very good, Emeran. So let's give some resources to listeners. So the Mind-Gut Connection, your book sounds like a very good place to start. I'm sure it's on Amazon and other places. Any other references or places that people should look when thinking about their own health or wanting to learn more about this subject?
Yeah, so our website, so it's emeranmayer.com, is a good place because you can also, once on the website, sign up for our newsletter. We have a series called the Mind Gut Conversation, where I interview, just like you interview me now, as I interview prominent people in the field. Facebook page at emeranamayer.com. At Emeran A. Meyer. So those three, I would say, the book obviously now out in paperback has a lot of the information, the basic information, and the update on what's going on will be found on the website or on the Facebook.
Okay, well, very good. Emeran, thank you so much for coming, and it's been there's still millions of more things to ask you, but this was a great start, so I appreciate you being here.
Okay, Rich, good talking to you.
You have been listening to Almost Here, Around the Corner Future Technology podcast with Richard Jacobs. Subscribe to this podcast, post a review to discover more future technologies that are poised to transform our lives for better or worse, such as Bitcoin, artificial intelligence, 3D printing, blockchain, virtual reality, and more.